Well, thank you so much for inviting me, and thank you all for sticking to the end. So I'm Melissa Spencer, and my lab studies genetically inherited neuromuscular disorders, of which there are 500 genetically defined forms. I did start out as a kinesiologist. That was my undergrad degree. So I have a warm place in my heart for exercise physiology, and I actually find that a lot of the work that I'm doing now in the disease state often plays off some of the lessons I learned way back in the 80s as a student at UCLA. So these neuromuscular disorders, they fall into some broad categories that were initially defined by their clinical presentation, but now have been stratified by genetic lesion. And the advantage of studying these monogenic disorders such as these is that you know the underlying cause, and that makes studying disease states a little more easy. So we study both Duchenne and limb-girdle. My talk today is going to focus on Duchenne, and we've already heard an introduction. This is the most common lethal childhood disorder, and it's due to mutations in the DMD gene, which encodes this large yellow gold protein here, shown at the subcercolemmal membrane, so on the inside of the cell, but attached to the membrane through actually interactions with phospholipid. And it also interacts with this complex called the dystrophin glycoprotein complex. So dystrophin is a scaffold for many different proteins. It also interacts with microtubules and actin and intermediate filaments. So it's a very important protein that is concentrated at costamers. And so let me just go back one. So in the absence of this dystrophin protein, when you have mutations in the DMD gene, you lose this entire complex, and you lose an interaction between the extracellular matrix and the intracellular cytoskeleton. And this causes the cell to become, to have an impaired sarcolemmal membrane, so it's compromised, and exercise and forces on the membrane cause rupture. So in order to describe what I'm going to describe, I had to overly simplify things. So dystrophin has three main domains. It has an actin binding domain at its N-terminus. The C-terminus interacts with the dystrophin glycoprotein complex, and it has this raw domain that consists of 24 different spectrin repeats, and these are represented by these balls. And it's the spectrin repeats that are thought to give the molecule some spring-like properties, and also it's those spring-like properties that help protect the sarcolemmal membrane. But the business ends of the molecule are the N and C terminus. There's an allelic disease called Becker muscular dystrophy, and in this case, you have mutations in the DMD gene that spare the reading frame. So in Duchenne muscular dystrophy, mutations tend to lead to loss of protein production. Becker muscular dystrophy involves mutations in the exact same gene, but because of the reading frame is spared, you retain those business ends of the molecule. And these Becker muscular dystrophy patients have a much milder phenotype than in Duchenne, and it's believed that it's because this raw domain is somewhat indispensable. So you can take out regions of the raw domain and still have some functionality. And this is the basis for how the current gene therapy trials have been designed. And so Jeff Chamberlain, over at least a decade, looked at this molecule and tested different domains and different constructs to figure out how much of this 14 kb mRNA he could eliminate to fit into an AAV vector. So it needs to be about 4.8 kb to fit into AAV to be able to deliver it to the muscles. And so this idea has been the basis for many other therapies as well. the microdystrophin retains about 30% of the protein. So it is not as functional as the wild type, but boys who are receiving this therapy are doing pretty well. So this idea of reframing the gene has also been the basis for antisense oligo-mediated exon skipping. And we have 3 approved drugs that are based on this idea. And so antisense oligos are just small chemically modified oligos that are administered through the bloodstream. And they act on the mRNA, the pre-mRNA. So in this case, you'd have a DMD patient who has a mutation in which once you have exon splicing, you create an mRNA that is non-functional, that has a premature stop codon. But when you remove or eliminate the exons that are interfering with the proper reading frame, as represented by these rectangles, you can then get a functional mRNA and a dystrophin that retains the business ends. Each patient will have a different dystrophin species that's created because of their different underlying mutations. And so with this idea in mind, we started to think about how can we improve upon these therapies. We know that there are 3 FDA-approved antisense oligo-mediated exon skipping drugs. They're all creating about 1% dystrophin, which is certainly having an effect, but it's not really ideal. And those drugs have to be administered weekly. So they're infused in an infusion center or through a port, and they only can skip a single exon. So we know that in Becker muscular dystrophy, patients can have different sizes of deletions of this raw domain. And we know that exon 45 to 55 is the mildest Becker muscular dystrophy phenotype. So this is a deletion of 11 exons. And in studying all of these different patients, this appears to be the most functional dystrophin species. And there's patients diagnosed as late as their 60s. We also know that this exon 45 to 55 region is a hotspot for patient mutations. So what I'm showing you here is the frequency of mutations along based on the exon. And you can see in this region, it's about 50% of mutations map to the 45 to 55 area. And so we sought to, instead of using antisense oligos, to use CRISPR, gene editing, to try to mimic this exon 45 to 55, knowing that it is the most mild BMD mutation, and that if we can develop this therapy with these pair of guides, we could have a therapy that would be applicable to about 50% of patients. And that is a huge improvement over what's going on right now with the antisense oligos, where each individual oligo has to go through the regulatory process, and each individual oligo can only skip 1 exon. So this is what our platform looks like. I'm representing the guide RNAs as these bolts. And we targeted our guide RNAs to the very furthest out region of the intron. So the exons are represented by these blue rectangles. Lightning bolts are the guide RNAs, and you can see we're very far out, primarily because we wanted to encompass as many patient mutations as possible. So when a mutation occurs in a patient, they can extend deep into the introns, and these introns are quite large. So when our guide RNAs cut in the intron, then the non-homologous end joining system of the cell will take these 2 introns and put them together to make a chimeric intron and delete about 708 kb. This is a huge deletion. We published this in 2016, and it's actually happens not, we were surprised at how well it happens. And so we initially validated the platform in iPSCs that were differentiated to either cardiomyocytes or skeletal muscle cells, and showed that we could take an out of frame mutation and reframe it with our guide RNAs and Cas9. And what's shown below is Western blot. And the dystrophin that's produced is smaller because we're taking out 66 kilodaltons of the protein. And then we had to generate a humanized mouse because we want to ultimately translate this platform. And the guide RNAs are very specific for the sequences. And so because our guides are directed to the human gene, we needed to have a mouse that had the human gene. And so we got the mouse from Jackson Labs, actually, that was created in the Netherlands that had the full-length DMD gene in a backbone. We mutated it with CRISPR to remove exon 45, and then we had to cross it to dystrophic mice to generate a mouse model that we could use for our studies. And we're attempting to deliver our platform in a variety of ways. I'm not going to talk about most of them today. We're doing ex vivo editing and engraftment, and this is mainly April Pyle who's doing this. We've spent a lot of time developing non-viral nanoparticles of many different types, not just lipid, but also silica and other types and other nanomaterials. But what we've ended up focusing most on right now is using adeno-associated viral vectors. These vectors are now being put into human patients through systemic delivery. I'm not going to say they're extremely safe. They actually have a lot of issues, which we're going to try to address, but they're highly efficient. And so in order to use AAV to deliver our platform, we need to actually have two AAVs. Because of the size of Cas9, it just reaches the packaging capacity of the vector. And so using a muscle-specific promoter, which is important, we have one vector that is CK8 driving Cas9, and another vector in which we have our two guided RNAs driven by RNA promoters, and we stick in an mCherry just so we can track the efficiency of transduction. And when we use AAV with our platform to delete exons 45 to 55, we get pretty efficient transduction, especially of the heart. So you can see I've stained with, not I have stained, Courtney Young and Michael Imami, my students, have stained for dystrophin, which is present at the sarcolemmal membrane, and you can see it here, and in both the heart and the diaphragm. And we've also done Western blotting to quantify the levels using also a standard curve, which isn't very good right here. But this is a very large protein, very difficult to Western blot. You can see in the heart, in this case, we had about 60% dystrophin. And just to remind you, what the antisense oligos are giving is about 1% dystrophin. But when we do skeletal muscle, we have less dystrophin. We get between 2 and 24% depending on the muscle. So I'm showing you here diaphragm and tricep. You can see the diaphragm is giving higher levels of dystrophin than the tricep. And we think this efficiency is really not the efficiency of the platform in different muscles, but rather the efficiency of transduction by the adeno-associated virus. And these are things we're working to improve in my lab. But in spite of the low transduction of skeletal muscle, when we did functional testing, we actually saw improvement. So we developed a wire mesh test where the mice hang from a grid. On a swivel, we just flip it over and we see how long they can hang. We used open field and looked at the distance traveled and also the rare events, and we've also been doing pulmonary function. And we do see benefit of even a small amount of dystrophin. But these studies are done under optimal conditions. They're done we are dosing very young mice who are transduced much easier. And we know that we really have to improve our platform and all aspects of it before we can ever go into patients. And that's our ultimate goal. So how can we improve our platforms? Well, one is to just improve the CRISPR itself. And we've tried designing various guide RNAs to delete various segments of the gene. All of these are would create dystrophins that are highly functional through modeling. And you can see if we use these different guide pairs, we create different sizes of deletions. And what we found is that if we just delete one single exon, which is about 1 kb in this case, so this triangle here represents our deleted exon 45 that we created in our mouse, we see decent efficiency. But as we increase the size, so here we're going from 1 kb to 100 kb, and that's this bar here, we drop the efficiency by about half. But as we go along, oopsie, I guess I took that slide out. As we go along and create larger and larger deletions, including the one we're interested in translating, which is this red bar, the efficiency doesn't drop off that much. It is a little less efficient, but comparing a 400 or 500 kb deletion versus a 700, there isn't much of a difference. So this results told us that we're probably not going to want to create these other deletions instead of this exon 45 to 55, because we want to encompass as many patients as we can with a single pair of guides. We will consider in the future, but for right now, we're going to take this forward. So it seems that the best way that we can improve our therapy is by improving the AAV vector and some of the conditions under which it will be delivered. And so we know now through these trials that have occurred that the systemic delivery of AAV, high levels of AAV, comes with a lot of problems. One of which is that it can only be a single dose therapy because of the immune response that develops. And so we know that not all myonuclei are edited. So if we use AAV, we deliver it, we see some correction, and we haven't been able to really quantify the amount of editing in a muscle fiber. But we do know that there's loss of edited nuclei over time. So even if you have a pretty good transduction and good editing, you're still going to lose them over time because of the non-corrected satellite cells that will replace those nuclei. And especially in a dystrophic muscle that hasn't reached the threshold of dystrophin that's needed to protect the membrane, you're going to lose those corrected nuclei pretty quickly. And there's evidence for this in the literature. There were 2 studies that looked at mice over 18 months, and then a recent paper by Jeff Chamberlain's lab that showed that over 18 weeks, you've already lost a lot of the edited nuclei in skeletal muscle, but not heart. So if you look on the left side, this is a 4-week time point, and you can see he's put all these different muscles here, and it's a little hard to see on my slide, but you can see that it's much lower dystrophin than what he sees in the heart. The heart is very well targeted by AAV. But also the heart is not experiencing as much, well, I can't say that, I'm gonna take that back. We don't know why. We don't have all the reasons why, but we know that the heart is very well targeted. And so you have a lot of dystrophin there to begin. And maybe it's spared because you reach that threshold that protects the membrane, whereas you don't reach that threshold in skeletal muscle. After 18 weeks, you can see he's lost the dystrophin from many of the muscles, but he's actually increased the editing in the heart. So one of the ways that we can overcome this issue is by targeting muscle stem cells. So rather than targeting myonuclei, which is what most of the AAVs do right now, we can target the muscle stem cells, which asymmetrically divide, they replenish their niche, and they go on to repair the muscle. And so we expect if we're efficiently targeting muscle stem cells, we could actually increase our corrected nuclei over time rather than decrease. And this is a project of my graduate student, Michael Imami. And so where Michael started is there are some reports in the literature that AAV9, this is one of the serotypes of AAV, efficiently targets muscle stem cells. So he decided to check for himself. And he looked at neonates and he looked at adults. He isolated the muscle stem cells and he sorted. So quiescent muscle stem cells are CD34 positive, alpha-7 integrin positive. So that's his population here. But he also looked at these other populations like the alpha-7 integrin positive, which are probably myoblasts. And after dosing in neonates and adults, sorting the muscle stem cells, getting rid of all the Lin negative cells. He then looked to see whether he got editing. And what he found is that in neonates, he could see a tiny bit of editing. So this is based on a PCR assay we developed to look for our deletion. But when he looked at the mice that were dosed as adults, he did not see any editing. And so we think what's happening in the literature in these reports is, first of all, they're all done with reporters. So they could see the editing on the reporter. They could get GFP positive cells that then divide. And they could be looking at activated cells. We don't really know why those reports show such a high level, but we're convinced that we really need to develop better AAV capsids that target muscle stem cells. And so that's what Michael's doing. He created a peptide display library where he switched out within this capsid a 7 amino acid sequence random. So he generated this library. And it is the capsid that determines the serotype, and it determines the tissue specificity, because these all bind to some receptor, usually a sugar on a receptor like the EGF receptor, for example. So he created this library, and then his workflow has been to transduce mice systemically, sort the cells, and then take different tissues to look, including muscle stem cells, to look for his library. He then enriched it, and that's where we are now. He's done 2 rounds. We're also looking for AAVs that, for example, don't target the heart, because some of the dystrophies, you don't want your AAV going to the heart. And we're very interested in detargeting the liver, because the liver is a source of a lot of these serious adverse events that have occurred. This is showing just that he's detecting his library in skeletal muscle based on this band here. So he knows exactly where the insert is. And so he can PCR around this, he can sequence around it and know what the peptide is within his capsid. And he's also found that he can detect some of these capsids in muscle stem cells. And that's where we are now with this. So as I said, one of the limitations of AAV as a gene delivery vector, especially for CRISPR, is its single dose. And as of now, we don't think any of the AAV serotypes target muscle stem cells, but we're looking for some that will. And the second is that there have been some serious adverse events in some of the trials. Some children have died in myotubular myopathy studies, so babies. And some Duchenne, at least 3 Duchenne boys have had some serious adverse events, one almost dying. And so these were sort of unexpected, because most of the studies have been done in mouse and really had not looked at the immune system sufficiently. And we don't really even have a nice way to screen. And so my lab, I'm not going to have time to talk about it, but there's a big effort to unbiasedly examine immune responses, both innate and adaptive immunity, looking at complement, because this seems to be a big problem, and seeing how changes in innate immunity affect subsequent adaptive immunity. And the goal ultimately is to be able to redose, because we know that if we can target some of these immune components, we'll be able to then redeliver and efficiently have efficient editing in a number of myonuclei, even if we never targeted muscle stem cells. And so I'm not going to show a lot of data. I'm just giving an example of sort of how we're going about it. So we are doing single and double dosing. We're then taking all PBMCs and doing single cell sequencing. And so you can see here on a single dose, we get a shift in phenotypes in almost every cell type we look at, including NK cells and monocytes. If we do a double dose, we even see a further shift. So if you're just looking here, for example, at CD4 T cells, you can see that they start here and they go all the way to, actually they go here and then back to these green cells. So we're looking at the cells involved in rejection, which is what happens after a double injection, and looking at the transcriptomes of those cells to understand what are the mediators. So in summary, I think it's cut off, sorry. Duchenne muscular dystrophy is amenable to CRISPR-Cas9 gene editing because the gene or the protein can tolerate large in-frame deletions. And then although AAV is very efficient for delivering CRISPR, we are not where we need to be. We even need to improve baseline screening as well as other safety factors for gene replacement strategies such as AAV microdystrophin. We've identified muscle stem cell targeting AAVs that may help overcome some of the limitations, and we're looking for immune modulators in mice and then comparing to human that may be targeted to dampen the response to AAV. We think that comparing our results between mouse and human will tell us what we can use or what markers in the mouse are useful models of the human immunity, because we know they're not going to be the same. So the work of the CRISPR platform, the developing of the platform, was done by Courtney Young, and this is a picture of her when she just entered grad school in 2014. She wanted to work on Duchenne because she has a cousin named Christopher who has DMD. And she's graduated, and she is now CEO of the startup that we spun out. This is my disclosure. So I'm a co-founder for Myogene Bio. Courtney is the CEO, and my other partner colleague is April Pyle, who is the co-mentor for Courtney. And we've all been working together now for about 5 years to really transform this to patients. And so this is a bit of a messy slide, but this is my lab. The two boxed people are the individuals who've done a lot of this work. April Pyle is a professor of MIMG at UCLA. And this work was funded by many sources, but primarily CIRM, the Muscular Dystrophy Association, and our UCLA Broad Stem Cell Institute, and now being funded by the DoD and NIH. Thank you.
Wow, thank you very much, Dr. Spencer. Fantastic presentation. Extremely important topic. We certainly have time for questions. Our first question is from Dr. Thomas Hawk at McMaster. Very interesting data presented, thank you. Have you interrogated where AAV6 is being sequestered in your mouse work, if not going to muscle? For example, is it all going to heart or liver or kidney?
Okay, so what happens with AAV is, so it is a protein shell that encapsulates a single strand of DNA. And that capsid, just like with coronavirus, with the spike protein, it finds a receptor, it enters the cell, and it uncoats. And the DNA that's inside circularizes, and it's in the nucleus, and it stays there. There's been a lot of studies looking at biodistribution of these different AAVs. Jeff Chamberlain especially has done a lot with AAV6. We're using right now AAV9, but we plan to change probably. We're using AAV9 because it was the first to be dosed systemically in patients with SMA. And those are babies that would have died had they not received this therapy. And that was the first systemic gene therapy to receive FDA approval. So we've been working with AAV9 because of the FDA's. I mean, we started because we thought the FDA would be more accepting of it because it had already been in patients. But with all of these serious adverse events, we're thinking that we may want to change. And we're also seeing somewhat low transduction of muscles. So AAV6 does better than AAV9. We're going to be trying some other serotypes, including those novel ones. So there's not sequestration so much, but there is a propensity for the different AAV serotypes to transduce different types of tissues.
Right, thank you. We have time for one more question. I don't see any list. Oh, wait, one moment. Oh, yes, there's two more. Another question, are there any vectors apart from Oh, sorry, it says my connection's unstable. Can you hear me, Dr. Spencer?
Yeah.
Are there any vectors apart from AAV that can be substituted for CRISPR-Cas9 transduction?
Well, we've tried to use non-viral vectors. We've spent years, I mean years, just lipid nanoparticles, silicon nanoparticles, polyritaxin-based, nanoparticles. They can get to muscle, but they can't traffic through the extracellular matrix. They cannot get into the cell. We would love to not use AAV. We're still working on it. But other than AAV right now, there's really no efficient way to transduce muscle by systemic administration.
Mm-hmm. I've gone 1 minute over, but because of the interesting questions, but there's just one last question. Anthony Shime here at York. Have you used your CRISPR approach on mouse embryos to correct the defect?
No, but it's been done. So Eric Olson published a paper in Science. It was one of the first to actually do this in Duchenne. We haven't done it. I mean, ultimately, I think, let's say in 30 years, when we can do postnatal screening or even prenatal screening, and we can identify patients with DMD and treat them early, that would be ideal, or at least at birth, because the sooner the better. But there are a lot of things to do before we get to that point. And it's also, sometimes you don't see the mutations, they're hidden. They're splicing mutations or intronic.
Right.
So, but hopefully sequencing methods by then will be much better. They're already great, but we need to improve. And that would be a dream, to be able to never ever have another Duchenne boy, because it is a really devastating and awful disease.
Yeah, yeah. And I see your company is focusing on the 45 to 55, which is
right now, yes.
Yeah, which is what percentage of the Duchenne, of people with Duchenne does that represent? About 20%?
50 to 60% of patients
50 to 60.
can be encompassed.
Okay.
Yeah,
Well,
and it's not just the exons. It's actually, because we've gone out way far in the introns, we think we'll be able to get more patients than if we targeted just the immediate flanking regions of those last exons.
Right. Incredibly important, fantastic presentation, and thank you very much for sharing your work, important work.
Thanks for having me.